Guys, I want to talk about cell membrane transport today. So this is going to be things moving in and out of a cell through that cell membrane, that protective barrier that we talked about before, and it's selectively permeable, right? And this is going to be quintessential to maintaining what we call homeostasis. And we're going to talk more about that later, but homeostasis of a cell or of an organism as a whole. Okay, so what I have here is a nice simplified plasma membrane. Okay, so I have the hydrophilic head right here, and then the hydrophobic tails coming off, and then I have a hydrophilic head down here and hydrophobic tails coming off. Remember that the tails like to hang out with one another and the heads like to hang out with one another. Okay, so in this diagram, perhaps underneath here is the inside of the cell and outside is on top. That's typically what you're going to see with your textbook, but you really should make sure that everything has a label. So it might say intracellular and extracellular, or inside and outside. Okay. Now, things can move into or out of a cell through two different major categories of movement. Okay, you have passive transport. So passive transport, when you think of something passive, you think of something kind of just sitting there. It's not spending any energy. Okay, so passive transport is going to not require any energy. It's just free movement, basically. Okay? Whereas active transport, when you think of something being active, you think of it using a lot of energy, and that's exactly the case here. If something is using active transport, you are actively adding in energy in order to make something move. Now, passive transport has another name. We call it diffusion. Okay, and basically, there's still energy in this system, right? Remember that all of these particles are moving. So I don't want you to think of passive transport or diffusion happening with zero energy in the system. It's just that you're not adding any energy. All of these particles are moving, right? They have a kinetic energy. So like if I were to stick a thermometer in here, it would have some kind of a temperature that you could measure, because remember, that's just measuring all that kinetic energy and averaging it. Okay? So molecules like to move from areas of high concentration to low concentration. If there's one thing you take away from this, let it be this. Things like to move from areas of high concentration to areas of low concentration. Okay, so if you have a lot of stuff all clumped in a small space, like in this upper left quadrant here of this glass, well, they're going to naturally start to spread out. And so that's what's happening here. You're not stirring this around or anything like that. You're literally just letting it sit. Okay? And it's going to eventually spread so that each particle has some distance away from other particles. Okay? And usually that distance is going to be pretty consistent throughout. You could average that distance, right? Now, keep in mind that these particles are still moving. So what I'm looking at here is really just a snapshot in time. And we don't typically see these particles, right? Because they're not typically things that are big enough to see. But I'm going to show you some examples of this in the real world that you have seen to kind of give you a nice idea of what it looks like. So what I have along the bottom here is I have a high concentration of this solute, whatever that solute is. It's a hexagon, so maybe it's a sugar. Okay? It's a high concentration over here. As I get to the middle, notice that some of those sugars have started to kind of separate and they're starting to spread out a little bit. And then eventually, with enough time, so time is my x-axis here, and I'm increasing as I go to the right, eventually it's going to pretty much equalize. Okay, so the overall concentration is going to be lower as it spreads out. Okay, because if you remember from chemistry, concentration, and I'm just going to abbreviate that C, even though that's not really proper, but concentration, remember that sometimes you guys would use molarity to calculate concentration. It's just the number of things, whatever those things are, in our case it's going to be solutes, over some volume, right? So if you put, let's say, 30 grains of salt into an entire ocean, you're not even going to notice the salt, okay? But if you put it into a small glass of water, you'll notice it much more easily. Okay? So things are going to move until they reach what we call an equilibrium. Okay, so once it's like kind of settled and it's nice and spread out, we call that equilibrium. Okay, equilibrium. I'm just going to abbreviate that. That's going on right here in this rightmost picture. And this requires no additional energy. Once again, I didn't have to stir the thing around. That might help. It might make this happen faster, but I didn't need to do it. Okay, so maybe I put a big old clump, like a sugar cube right here. This is a sugar cube, and then slowly it starts to dissolve, and then it spreads out, right? You guys have seen this kind of thing happen. Now, here's another example. This one's more visual. Okay, you can take a simple glass or beaker of water and drop some food coloring or some ink, anything that, stands out like that and is hydrophilic, and it will start out in one small clump. So here I have it kind of clumped up here. Obviously there's some outside, and that's fine. Okay, but it's mostly clumped. So this area would be high concentration. Okay. And then you'll notice that in the middle, so here I have time, this is the same beaker just over time, or I should say flask, over time. Okay. And over time it starts to spread out some more, and then eventually it may look like this is still pretty clumped, and it is clumped. But notice that we're starting to see some blue at the bottom, we're seeing some at the top, we're really seeing a lot more spreading out. And so if you let this sit for long enough, you would eventually get this being a nice, clear, relatively clear blue, kind of like this, but all the way throughout. Okay. And we're going to do a demo to show this, and we're going to do a lab, so don't worry if it doesn't quite make sense yet, it will. Okay. But simple diffusion, I want you to start thinking in the context of cells now. So this is a great visual, and it's absolutely how this works. However, I want you to start thinking of cells, because that's really what we care about in AP Bio. Okay, so with simple diffusion, particles are going to go through those phospholipids. Okay, they're going to diffuse across without any extra help. Now remember that these phospholipids can't let just anything through. They have to be small things, and they have to meet certain criteria. Okay? And so that was all talked about in the last lecture, and we're going to solidify it here. But only molecules that are permeable to the membrane can pass right through. Okay? So that means only things that can make it through can do this easy thing called simple diffusion. Because otherwise, you need some help. Okay? So not much can do this. It really is a pretty limited list in terms of cells that we care about. Okay? But you'll notice that in this diagram, the same thing is happening. I have a really high concentration here, and a really low concentration here, because there's nothing over here right now. And remember that things like to move from high concentration to low. So all of these little blue hexagons, again, maybe they're sugars, but probably not in this case because they're passing right through the phospholipid. Okay? So whatever this thing is, it's going to pass right through. So over time, here's a time axis, we're going to see that some of them are going to make it through until eventually they're going to roughly equalize. Now in this case, we have the same volume. Okay, so like this amount of space is roughly equal to this amount of space. And so therefore the concentration is going to have the same amount of solutes roughly. So if we counted up all the hexagons, it'd be about the same. Okay, but just keep in mind that sometimes the volume is much bigger on one side, and we're going to practice with that later. But sometimes the number of, in this case, hexagons won't be equal on each side, even if the concentration is equal. And you kind of got to wrap your head around that. Concentration is what we care about when it comes to diffusion. Okay? So very few things can make it through the phospholipid. They have to be small, they have to be nonpolar, because they have to get through all those tails. It's really hard for most things. So sometimes things need a little help. Okay? So ions and some other small molecules can pass through a membrane. It's still a form of simple diffusion, but they needed a little help. They needed a protein. Okay, so this protein is just a channel. I want you to just picture it as a hole that only some things can pass through. Okay, so it's not letting everything through. It's not letting water through, for instance, in this case. It's just letting this yellow solute, whatever that is, pass through. Because I needed some help as a yellow solute, because I needed some help to go from the outside, the extracellular fluid, to the inside, the cytoplasm, I needed some facilitation, some help. So we call this facilitated diffusion. So remember, diffusion is still passive. Okay, so this is still passive. I didn't add any energy to the system. So that's not the form of help that I used. The form of help I used was right here, this channel. Because without this channel, there'd be no way for those yellow solutes to make it in. Okay, so you're going to see things go in and out of cells through channels a lot. And so we're going to practice with it. But for now, I just want you to picture it as a whole that is very specific about what things it can let through, rather. Okay. So when you start thinking about channels, I want you to think about specificity, as I just was talking about. Okay, so sometimes it helps to think about the channel being charged. So if this channel, this protein itself, this purple protein, is mostly negative, then it's going to push away any kind of negative particles. So negative ions aren't going to want to make it through here. But positive ions will get attracted to it, and then they may be able to find their way through. The exact opposite is also true. Now, I don't need you to memorize the fact that some of these channels are negative and some are positive. I just want you to think about the fact that these guys have ways of regulating what makes it through. Okay? So, we start to see this when we start talking about specific ions that are critical to cells. So we have sodium ions, we have potassium ions. Okay, and they need to go inside and out of cells and through other membranes. Okay? And so I really need you to just start picturing these as being specifically sodium channels and potassium channels. Okay, they can open and close, so sometimes you want to open one and close the other. Sometimes you might want to have both open. It really depends on what the cell is doing. Okay, but they are independent of one another. Okay, and we're going to practice a lot with this. But in the context of passive transport, we talked a little bit about this in the last lecture. Water will also move. Okay, so here in these diagrams, I have a semi-permeable membrane. That's what this dotted line is, this dashed line rather. Okay, and only water can make it through. Okay, water is able to go in and out of most cells pretty freely. Okay, so water is going to move towards areas of low concentration. So that's low concentration of that molecule, whatever it is. So if I'm looking at water, water is going to move from high concentration of water to low concentration of water. So that's what's happening across these two diagrams. But water is a little special, and we're going to talk about that later because it sometimes does the opposite, and that's really confusing sometimes. Okay, but when water is moving, we call that osmosis. Okay, you guys have heard that term before, I'm sure, but osmosis is just passive transport, but specifically water. Okay? Now, because we now have two sides of every situation, we have the inside of a situation and the outside. So maybe this is a cell. We have the inside of the cell and we have the outside of the cell. We need a way to talk about concentrations more easily. Okay, so if you look at this little diagram right here, there are a lot more solutes, a lot more of these purple dots outside of the cell. So here's my cell, and the outside has a ton of these dots. Okay, the inside doesn't have very many dots, but notice that the outside also has a much larger volume. So remember that concentration is the number of solutes over your volume. So you really have to take both into account. Sometimes it'll look like the outside is just so full, but sometimes the inside will have the same concentration. What really helps is to look at the distance between particles. So if you look at the distance between particles, you can see that overall, there's a little bit more space inside of the cell in this case. Okay, so the area that has a lower concentration of solutes is called hypotonic. And the area that has a higher concentration of solutes is called hypertonic. Okay, so if you are really, really squished in there and all of these particles are moving and they're all bouncing into each other like crazy, like this outside situation, we would call that hypertonic. Okay, if there is something that is hypertonic, it has a higher solute concentration, that means that there also has to be something in the system that is hypotonic. It's kind of like light and dark. You can't have light without having dark, and you can't have dark without having light. Okay? So, these guys, these purple solutes, I'm going to clear off my drawings here. These purple solutes really want to get inside of the cell. They want to move from high concentration to low concentration, right? Just like all things like to do. But sometimes you'll have a situation where the solute can't get in. Okay, so there's all of that potential there. These guys really want to get out. Okay, so here I have 300 and 600. Just picture that as a concentration. Things aren't usually 300 or 600 molar, but think of it as a concentration. Okay, so things want to go from high concentration to low concentration. Now, unfortunately, these purple ones cannot go into the cell, and that's a shame. Okay, the environment is hypertonic, so it wants to equalize. It wants to reach equilibrium, become isotonic. We'll talk about. Okay, but it can't. So something else is going to happen. Okay, so in this case, water is able to move into and out of the cell. Okay, and so basically what's going to happen is this low area, this low concentration area inside of the cell, well, it wants to equalize the concentration too, but the purples can't come in. Okay, the purples can't come in. So my number of purples per unit volume, the number can't change. The number of purples can't change. It's stuck. Okay, well, what can change? How can I get this concentration to be bigger? Well, I have this volume here. So what the cell will do, and it doesn't do this actively necessarily, okay, but this is just natural, right? I'm not adding any energy. It's going to try to decrease the volume. It's going to try to bring down the volume. And how's it going to do that? Well, water is going to leave. Okay, so water is going to leave from the inside of the cell and go outside. And that's going to have two effects. Okay, so if water goes from the inside of the cell to the outside, I have now decreased the volume of my inside of the cell, and I have increased the volume of my outside of the cell, right? Because water went from the inside, therefore it decreased inside, and it went to the outside, so therefore it increased the volume of the outside. So that means that my concentration of the inside went up because I have the same number of purples, but I have a smaller volume. Okay, so it's just simple math. Remember that concentration is just number over volume, number of solutes over volume. Okay, so because I decreased the bottom of the fraction, that means that think of it as if I had a volume of 4 and I had 5 solutes. Okay, so I had 5 solutes over a volume of 4. Okay, so my simple little division here would be 1.25, right? So that's my concentration. Okay, so 1.25. But if I decrease the volume down to 2 instead of 4, like I'm doing here, well, now I'm going to have 5 divided by 2, so now my concentration is going to be 2.5. So my concentration just went up. And the exact opposite is going to happen outside of the cell. So the outside of the cell was very hypertonic, okay, because it has a higher solute concentration. However, as more water goes out, my concentration is going to lower because I'm increasing the volume of the outside. And I'm dividing by a bigger number. So I'm cutting my pie into more pieces, so each piece has to be smaller. Okay, and we're going to practice a lot with this, but I need you to really start thinking about this. Okay. So if the solute concentrations are equal, you may think that water movement will stop. Okay? And water movement kind of will stop as a net. Okay? So the net movement will stop. Things will kind of calm down and sit for a while. But just remember that things are still moving in and out, just at an equal rate. So things are still moving, water's still moving out, and water's still moving in, even if they're nice and isotonic. Okay, isotonic means that the inside is equal in concentration to the outside. But for every one particle of water that leaves, one particle of water comes in. Okay, so it's equal. So even though it's at equilibrium, that does not mean that there's no movement. It just means that the movement is equal. Okay, so in this situation here, right here, what I'm circling, I want you to go ahead and try to identify which part, whether it's the inside of the cell or the outside of the cell, is hypertonic. Okay, so I'll give you a quick second. Think about which one is hypertonic. The way I like to remember this is hyper. I think of all those particles being all squished together. They really want to get out, so they're all nice and hyper. Okay, they really want to get out and get some freedom. Okay, so you should have realized that these guys on the inside of the cell are really squished together, and they also have a value here of 300. Okay. They're hypertonic. So the inside is hypertonic, therefore the outside is hypotonic. Okay? So if I want to equalize those concentrations and the purples cannot move, Where's the water going to go? Okay, I want to equalize the concentrations. The purple can't move, so the only thing that can move is water. So what's the water going to do? Well, as you can see in this diagram, the water is going to go into the cell. Okay? It's going to make the cell bigger, so I'm going to increase the volume, and that will decrease the concentration. Just remember that volume and concentration have a disproportionate relationship. Okay? If you increase one, it decreases the other, and vice versa. Okay? And that's just because of this little equation right here. Okay? Down here I have the exact opposite. I have a hypotonic inside and a hypertonic outside. Again, purples can't move, so the only thing that can move is the water. And so the water is going to move where? It's going to move outside from the cell, okay, to try to equalize those concentrations. Okay? Now this becomes really important when we start talking about cells. Because remember, cells are typically going to be in a solution. I mean, you can think about blood cells because that's the easiest visual, but your blood cells are in your blood, and your blood is all that liquid, right? So you have them inside of solutions. And so you don't want too much water going in and out of a cell. You want it to be just right. Sorry about that. So I have here a cell in a hypotonic solution. Okay, hypotonic solution. So just think about that for a second. Which one is low concentration and which one is high concentration? Okay, just think. So if it is a hypotonic solution, that means the outside is hypo. That means it's a low concentration. And therefore the inside is a high concentration. It's hypertonic, right? So if things can't get into and out of the cell other than water, then water is going to do the job. And water is going to flow into the cell, just like we saw before. Okay, that's what's happening in this top part right here. Water is flowing into the cell, which can be fine until a point, until it's not fine. And then you can actually have a cell rupture. So if your concentration gradients aren't just right, your cells can literally explode. Okay? So that's what's happening here. We call that being lysed. Okay, lyse comes from the root for cutting. Okay, it's more like a pop though. Okay, but some cells actually rely on this. So if you look down here, excuse me, if you look down here, you have a plant cell, and this plant cell has a big vacuole. Okay, and plant cells are a little interesting because they have a cell wall on the outside. And as you guys know, plants like to be very solid. They like to be rigid, right? Because they need to fight gravity. So they actually want some more water going in because that's going to allow the water to fill the inside and then kind of push against the cell walls. And that actually keeps it nice and rigid. So just understand that some cells really don't want water coming in and some cells really do want some water coming in. But any extreme is pretty bad, right? You don't want too little water or too much water in any situation, okay? And then I'm going to move over to the hypertonic solution. Okay, so if it's a hypertonic solution, which one is hypo? Which one is hypo and which one is hyper? Give you a quick second. So if the solution that it is in is hyper, that means that the inside of the cell has to be hypo. So which one has a high concentration? Which one has a low concentration? My concentration on the outside is high. Concentration on the inside is low. Okay. So if I want to equalize these concentrations, water is going to want to move outward. Okay, and that's exactly what we saw happening down here. Water wants to move outward to equalize this concentration. Okay, and when a cell does this, it becomes shriveled. Okay, so a typical animal cell will become really shriveled. Okay, it kind of looks like a raisin, because that's exactly what we do to make raisins happen. We pull the water out of it. So if you pull the water out of a cell, out of a grape, it's going to become really shriveled. It's the same idea. Okay? And then in the case of plants, they're going to be plasmolyzed, and basically they're going to be really, really weak and floppy. Okay, so when plants are dying, you start to see this happen. Okay? Now, isotonic, that's our final situation here. Isotonic, iso means same, so an isosceles triangle, right, has two sides that are the same length. Okay? So iso means same, and tonic is referring to the different solute concentrations. Okay? So that means that the amount of water going in is roughly equal to the amount of water going out. And generally for an animal cell, this is exactly what we want. That's normal. For a plant cell, that's going to be fairly flaccid. It's not going to have the rigidity that it really needs. Okay, so again, there's just some diversity in what cells need, and that's totally fine. Okay? So here's just another nice little visual about what happens when you put cells into different solutions. So here I have some blood cells. If you put them into a hypertonic solution, they're going to shrivel up because all the water is going to leave. If you put them into a hypotonic solution, too much water is going to flow in and they're going to burst. Okay? And then if you put them in an isotonic solution, it's nice and perfect. Think about it like Goldilocks and the Three Bears. This one's too high, this one's too low, this one is just right. Okay? Now, this becomes really, really important when we start talking about medical procedures because you don't want cells bursting or cells shriveling when you have a patient, obviously. Okay, so those of you who are looking into the medical field, just kind of keep this in mind. This is really quintessential to what you're going to be doing. Okay. Now, most tissues are going to be stored in what we call a saline solution. Okay? So saline refers to salt. Okay? So if you think about like saltines, the crackers, they're literally just crackers with salts on them, right? Kind of sounds a lot like saline. That's not a coincidence. Okay? So we put them in a saline solution of sodium chloride, so NaCl, and that is in fact just table salt. Okay? And that solution has the same osmolarity. Remember that osmolarity is really just the same as saying concentration. Okay? It has the same concentration. So I can put cells into this solution and they're not going to get hurt because it's nice and happy. Okay? I'm here in the nice isotonic situation. And so that's going to be what we use to rinse wounds because we don't want to burst or shrivel blood cells, right? Anytime that you're really dealing with any kind of injury, it's going to be a saline solution. Or if you are dehydrated and you're on an IV drip because you're dehydrated, they're going to put a saline solution into your veins. Okay, so that's going to be water, but also a little bit of salt. Because if they just put pure water in there, now you're causing the outside solution to be too hypotonic, right? So, just something to think about. So if you ever are in a hospital, like I was, and they put an IV drip into you because you're dehydrated, just realize that that is not just water. Okay? Just a little interesting little fact. It's a saline solution. Okay? Now this kind of takes us to the other side of the coin. This is active transport. Active transport requires energy. Okay? And you're going to see this term free energy, and we're going to talk more about that moving on into the next lecture. But it's nothing's really free, right? This is kind of a confusing term, and it just comes from where the energy ultimately comes from. But just think about cells as having energy in the form of ATP. Okay, so ATP you guys have heard of, it's the cell's currency. Passive transport doesn't need ATP, but active transport does. Okay? And this basically allows us to break the rules. This lets us move particles from low concentrations to high concentrations. Remember, that's the exact opposite of what they want to do. Normally things want to go this way, but I'm able to pump things this way. Pump. And that's by using energy. Just like if you have a pool pump, you're doing the same thing. You're using energy to pump water, move it around. Okay? So in order to make this happen, you really need proteins inside of a membrane. So here I have 3 different examples. Okay, I have simple diffusion, so things are just moving through the membrane. I have facilitated diffusion, which is things moving through the membrane, but they need a little help from a protein. And then I finally have active transport, which is stuff moving, and it requires energy because it's going against that concentration gradient. And this is a really nice visual. I'm going to get rid of my arrows here. This is a really nice visual because look at how few green squares there are on the outside of the cell and how many there are on the inside. So these guys want to leave, but I'm not letting them leave. Instead, I'm going to add even more. Okay, I'm fitting more people into this room. So they're getting more and more uncomfortable. They want to leave even more, and that's going to become really important. But as you can see over here, passive transport, has two categories we've talked about. Okay, we have diffusion, so just simple diffusion. And this is really hard to type or to write, but oh well. And then we have facilitated diffusion. So this is simple, and this is facilitated. Okay? Active, at this point, only has one kind, but these are both ways of moving things into and out of a cell. So I kind of like to think of it in terms of a flow chart. You want to move something? Okay, do you need energy? No? Okay, so then we're going to be on this side. Can it get through the membrane? Yes? Okay, simple diffusion. Can it not? Okay, we're going to need a protein. Okay, I need to move things into or out of a cell. Is it going against its concentration gradient? Yes? Okay, I need to add some energy. Okay? So protein pumps are going to become really important. Proton pumps, which are proteins, are going to become really important moving forward. Okay, so protons, think about hydrogen. Hydrogen has just a single proton and a single electron, right? Just a regular hydrogen atom. Now, if you take away the electron, so I take out the electron, I really just have that inside part, right? And now I got rid of a negative charge, so now I'm going to be positive. And so I really only have the one thing that was on the inside and a net positive charge. That's because it's a proton. This one proton has a positive charge. Okay, so anytime you see H+, it's literally just a proton. And you kind of got to visualize an atom losing its electron to really understand that. Okay, but we're going to start seeing protons being pumped in and out. Okay, so here we have a lot of protons outside, not many inside, and yet I'm going to make them fight against their concentration gradient. How do I do that? Well, I need some energy. So I'm going to spend some ATP. Okay. Now, vesicles allow us to move things as well, and that's just kind of a callback to the organelles that you guys read about over the summer. So I'm kind of going to skip over it for now, but just realize that anytime that you create a vesicle, you're really kind of creating a new area with new solutions. So now I might have hypertonic in here, hypotonic here, and this was still hypertonic as well, right? So now you have a lot of things going on. So just start thinking about it. You don't need to memorize anything at this point, okay? So your book starts to talk about endocytosis, and that is really important, but the big thing I want to talk about, really the only thing I want you to learn, is phagocytosis or phagocytosis, depending on who you ask. Okay, so this is when large things kind of get swallowed whole by a cell. So what happens is it gets usually bound on somehow, and then the cell membrane kind of lumps over it and then forms a little bubble. And then that allows you to take it into the cell while still kind of keeping it in its own little compartment. And we're going to talk more about that later. But exocytosis is the exact opposite. You put something in a bubble and then you make it leave the cell. Okay? Now, here's a nice little overall visual for endocytosis versus exocytosis. I just want you to start thinking about these as being cell membranes within cell membranes. Okay? Because that's really what this is. This almost looks like a cell in and of itself, but it's all inside of the real cell. Okay, so you have a lot of solutions going on here. Okay, and then this allows us to get rid of stuff or break stuff down or take stuff in that we otherwise couldn't get in. And we're going to talk more about this and we're going to practice with it, so I don't want you to freak out about it. But for now, that's going to do it for membrane transport. Remember that we have active and passive transport. And remember that passive transport breaks into just simple diffusion and facilitated diffusion. And that is going to do it for this lecture.